We are now recording, and you guys can see my screen, right?
Yep.
Yeah.
Okay. Hey everyone, we just wanted to make this quick video for you guys so that you could see exactly where to go to get all the information you need for each annotation and also how to write it in the notes section. So we're going to use Gene 29 as an example, and I think Andrew's going to start us off.
All right, so our first step is to find the start codon and determine that in the stop sequence. With normal genes, usually it's glimmer or gene mark singly that call the start, but sometimes you get ones that differ, and so this one does. Glimmer calls at 216688, and gene mark calls at 21667. So we don't necessarily know yet which start to choose from. What we're going to do is we'll blast our product through NCBI, and we'll compare that with the other blast using the other start codon. So Jackson has already done that, and now he's going to change it to gene marks call at 21667. He's going to press the calculator button and then does post validate, copies and pastes this, and then goes to NCBI blast and double checks to see. Okay, so we see first we look at the query. Where is that?
Query cover.
Yeah, so we look at query coverage for this one, and we compare it with our other one, and we see gene marks has a higher percentage. And also, if we were to look more into it, it's a 1-to-1 coverage with that. So that's how you can kind of choose between the two starts. So we're going to keep it at 21667 as our start. And our end is going to be 21404. It's important to note that usually 5' is your start, but because 29 is going in the opposite direction, 5' is our end.
Okay, and so now that we have done our start and stop codons, we move on to our coding potential. And so for this one, we're going to be looking at our gene mark analysis. And so when we pull up gene mark, we see all of the different humps. We're going to want to go down to our gene. So it'll start at 21667. And so we find that. So it'll be starting there, and we see we have coding potential here on the reverse strand. Remember, when you're reading gene mark, it has the 6 different reading frames on every page. But remember, these reading frames do not correspond to a specific frame that's designated +1, +2, +3 in DNA master. So you just kind of have to look at the forward going frames and the backward going frames and figure out which frame you're on. So we look here, and as we see over our gene coverage, we have this peak in coding potential. Remember, the graph shows coding potential. So if the line is, if you see a line, that means that there's coding potential. If there is no line, there's no coding potential. And so this coding potential goes over our whole gene that we see here. And so we're going to say that, yes, there is coding potential on this gene.
Okay, so next is our start choice source. That goes back to our start codon. What we just talked about, we're going to do gene mark, but because both glimmer and gene mark called 2 different ones, we're going to put both -GM. If we go back to our database on C pages that talks about documenting and how to do all of this, there's a graph that it gives you, and it shows that this is what you should do. So both -GM.
I can show where that is real quick. It's just in the document.
Okay, yeah.
Informatics.
Using this document is so key, and getting familiar with everything that's on the left is how you get more comfortable. This video hopefully will just give you a starting point, but yeah, so this is the graph.
Yeah, and just so you guys know how to use this graph, I just barely figured it out. But if it's called by glimmer, only glimmer, and it's selected by glimmer, you will put glimmer in. If you end up choosing a different start, so it was called by glimmer, but you don't use glimmer, you put glimmer CS. Same thing with GeneMark. If it was only called by GeneMark, you will put in GeneMark. But if you don't choose that it called, you put in GeneMark CS. And then both, you put in both, and then you put in GL or GM or both, depending on which one is right. So that's how you use that table.
Yeah, so in this case, we've done both GM.
Okay. Then next thing we have is Starterator. So if you guys remember, to get to Starterator, we have to use cphages DB, or no, just phages DB, my bad. And you type in Mandalorian or whatever phage you're working with, and you go down here to gene list. You click to view, and we are looking at gene 29. So we will go to gene 29 here, and that will open up another page. So we then come here at Starterator, click on the report. And this here is showing us the start sequences for several gene 29s for several different phages, all in the same cluster. So we can see here we have Mandalorian, and then these are the other phages in the cluster. And each one of these colored lines with a number are different start sites. And so what we're going to do is we're going to go all the way down here to Mandalorian, which is right here. So we see Mandalorian, and it says, it calls it having a start at base pair 21688, which you guys can remember was our original start site, but then we changed it, and that is start number 3. But then since we changed it to 21667, you can see here we're going to use start number 5. And so we can go back, we can see that here, that's start number 5. And so that's where the gene is actually starting. And so in DNA Master, we are just going to put SS because it's a suggested start. If we go back to Starterator and we look under the start information, so talking about start 5, it says that it is found in all of these genes in these different phages, and that out of all these times, 88.2% of the time, it is the correct start. So we know that start 5 is where the gene is starting, because that also lines up with our start position that we chose. So that's how you use Starterator. Now we're going to go over BLAST. So if you remember, we already put in our BLAST sequences, and we clicked on the first match that had the highest match. That takes us to this page. And there's just a couple different things that we need to put in to DNA Master. We have to start with the phage name that it matches. So let me see if I can edit here. It is Nuki. And then we also have to get the gene number. And so in order to find the gene number, you click here on the sequence ID, and that'll open up a new page, and you go down and you can see right here, it's gene 29. So we will go back here, and I believe it tells us to put GP29 or comma GP29. So that's the gene number. And then it asks for the database. And so if you remember, we're in NCBI. So we will put NCBI. And then after that, it asks for the query and the subject, query and subject. And so we can see the query and subject is 1-to-1. So we are going to put Q1 colon S1, big S. And comma, it is also asking for the percent alignment, I believe, which we can see here is 100%. I think it also shows it on this original page. Where's the original page? That's not it. Here it is. So you can see here percent identity is 100%. So we will put 100%, 100%, 0.00. And does it ask for anything else? Yes, it asks for the E value. And so the E value we can see here is 9 to the negative 56. So we'll put that 9e negative 56. And that is everything for the blast. And I just realized I put this all in the wrong spot. I put it after starter. So I'm going to copy and paste it real quick. Command X. That didn't work. Nope. This isn't working. So just know, I'm sorry guys, just know that this goes after blast start, not after starter. And we'll just...
If you do right click, that should let you copy and paste in DNA Master. It doesn't like all the shortcut commands.
Let's see that. Thank you, Tiana. Let's try that. Oh, you're a genius, Tiana. Cut. There we go. Right click, paste. Yes. Okay. And now we are on to gap. So I'm also going to talk about gap. So gap is the gap between the start of our proton, or not our proton, our protein, and the gene before it. So when the genes are going forward, so 1 through 26, we will subtract, for example, with number 1, we will subtract the stop of number 1 from the start of number 2. Is that correct? And that would be, that's not correct. Tiana, how about you explain it? I'm sorry.
So you want the gap that's before the gene. So for like gene 1, because it starts on nucleotide 1, there's no nucleotides before it, so there is no gap. And then for gene 2, you'll take the start, well, the end of gene 1 minus the start of gene 2, and you'll put down that gap.
Yes, my bad, guys. And in this situation that these genes are going backward, we need the gap between 30 and 29. So for 29 here that we're doing our gap, what we're going to do is we're going to go to 30 and subtract 2107, or what is it, 21707. The 5' end of 30, we're going to subtract that from the 3' end of 29, and that gives us 40. Does that make sense? I'm sorry, guys. Andrew, Tiana, does that sound right? Okay. And then one more that I'm going to talk about is longest open reading frame. There are several ways we can see that, but if you remember in Starterator, we see all these different start sites, and we remember that we decided that 5 is our start site. So that means there's start sites before 5s at 3, 2, and 1. So that means that there are other open reading frames that are longer than ours. So that means that for longest open reading frame, we are going to put no, because it is not the longest possible open reading frame. Okay. And now, Tiana.
Okay, so now we will move on. Yeah, so now we're going to move on to RBS. So remember, this is our ribosome binding site. So we'll go to DNA, and then we'll open our frames. And so then once we have our frames open, we'll click on the ORFs button, and that'll highlight all of our genes so we can see where our genes start and stop. And we'll scroll over to the gene of interest, so we'll go over to 29. And we'll select 29. You just kind of click on the box and you can make it bigger so you can see it better. So you want to highlight that whole gene and then click the little RBS button on the side, on the bottom. And so then this pulls up your ribosome binding site matrix. And so in this matrix, there's a couple things that we need to write down. So first off, we need to know what matrices we're using. So we'll write down our scoring matrix. So those we can find up in the top. And so we should all be using Kibler 6 and then the Carlin medium are our two scoring matrices. And then after that, we want to put in our z-score. And so we're going to be looking at the start that we have called. And so we called the start, if we look at the start position, we called the 21667. And so we want to be looking at that guy when we write down all of this information. So after we write down our matrices, we'll write down our z-score, which is this third column. And then after we get our z-score in, we'll move on to the final score.
1.6.
1.691. 1.691. So that's our z-score, and then our final score. So that's our -4, right?
Yep.
Yeah.
Got it right here. -4.699.
And so then it wants to know whether or not this start that we have chosen has our lowest, well, our highest final score. So if you notice, all of our final scores are negatives. So we want the one that's closest to 0. So the lowest absolute value. And so if we look in this case, that's our start that we have already assigned. So we would put yes. If there's another start that has a higher score or one that's closer to 0, then we would put no. And since we're not really using the ribosome binding site to decide between different starts, you can just kind of put that it's not applicable for choosing the start. So no, it's not the best score, but we will use that start regardless. In this case, we just put yes. And so then after the ribosome binding site, we move on to our function. And so the next 3 sub little notes are all about trying to assign the function and decide what it does. And so in this case, after we did all that, the best function that we found was membrane protein, but we will take you through how we got to that as we go through our CIF BLAST, CIF HHPRED, and CIF Synteny. And also for function, remember there is the list of approved functions. And so if it's not on the approved function list, do not write it down as a function.
Okay, so for CIF BLAST, which is our first supporting information for function section, we're going to actually go back to NCBI BLAST that we did earlier. Previously, we were looking at the BLAST for the gene specifically. This time, we're going to try to look and focus on the protein. So the first thing we look at is the function, and I believe it's membrane protein. That's what the function says on NCBI. Is that right?
Membrane protein.
Yeah, so we will put in, that's the function, membrane protein. The database is going to be NCBI. The phage name and gene number, that's the same thing that we put in last time. I think it's Nuki and gene 29. That you can find in NCBI. It's pretty easy. And then we get to database gene accession number. Okay, so what is this? Or if we go to the website, then we click on the phage, and we can find it in 2 places. Yeah, that's the easiest one. It's the sequence ID. If you go in specifically, you'll see it. But yeah, so just look for that. It's some kind of code. That's the accession number. That's what we're going to put down. So then we have percent alignment. Percent alignment is how much of our protein amino acid sequence is aligned with the query amino acid sequence. So this one's 1-to-1, so that's 100%. The way you determine this on HHPred, Tiana will go through and put that in, and that's all pretty straightforward. So.
Then we also put an E value, don't we?
Yeah, you also put that in.
Which is the same that we already found. So
Yep.
now we go to HH.
Okay. So now that we've done our CIF blast, we move on to HHPred. So you'll pull up HHPred and you'll copy and paste your protein sequence just like you do for blast. At the bottom, you're gonna have to select databases. And so there's 4 databases that we use. We use the protein database, which is PDB, and then our SCOP, and PFAM, and then NCBI conserved domains, or CD. And so those are the 4 that you want to use. Once you have those 4 and your protein sequence entered, you push blast or start, whatever it is. And then just like with NCBI blast, it'll blast it and it'll bring up all these results. And so then you look at your results and you want to find the one that has the highest probability and the lowest E value. And so if we look at these, our highest probability is 72 and our lowest E value is 13. And neither of those numbers is good. So for this gene, we just call off the search on HHPred here. So we'll say that it's no known function and that there are no, there's no matches with a probability above 90%. Because if it's not above 90%, you don't want to be using that to base your annotation off of. But let's say that this did have a match that was good. So we would want to put down the function. And typically if we look here at the name, it tells us what it does. So this is a pox virus entry protein complex. And so the function would be entry protein complex. And then afterwards we would want the database somewhere in HHPred. And then the phage name will also be typically in this name sequence. So here we have the name of our pox virus that we're looking at. And then if we want to look at the gene number, we can click on the hit, and that'll pull up a more detailed page about this protein.
Sorry guys, my internet is.
And so when that loads, okay.
Is this it?
This is it.
So real quick, Tiana, I think you put PFAM for the database instead of HHPred for this one specifically.
Oh, yeah, because this one's in PFAM.
Yeah.
Oh, yeah.
Yeah. So I'm not sure. I don't know where the gene is in this one. Anybody else know?
I've never seen this.
I haven't seen this either.
No, but it's good because for this gene specifically, we have NCBI blast is so awesome that it's okay.
Yeah, I'll say this one, it doesn't matter, but hopefully you'd be able to click on it and it would come up with more information and tell you where the gene was and all the other stuff that you need to know. And then the one big thing for this one is our alignment. So if we go back to our HHPred results, you have to write down the hit. And so that's just kind of this identifier in this second column that we clicked on earlier. So that's your hit that you write down. And then the percent alignment, you have to figure that out yourself. And so you go to the align cols. And so that's the number of amino acids that match between whatever you blasted and this this sample. So you take that number and then you go to DNA master. So this is 55. And in DNA master, you'll pull up the product and it'll tell you how many residues. So here it's 88. And so you'll do 55 divided by 88, and that will give you your percent alignment. And then you'll write down the probability, which is another one of the columns on HHPred. And so that's how you'll do your CIF HHPred if you get something that's above 90% probability.
Hopefully in the, for the most part, it'll be like this one, and it's less than 90%, so you don't have to put all that stuff. For CIF Synteny, that's the last one for this entire list that we're filling out. We're going to use Famerator. To use Famerator, you have to log in. Usually, you probably have to create your own, but for Mandalorian specifically, it's EA10. That's the cluster and subcluster it's in. You can find that information on Phages DB when you go to your Phage. We're going to unclick Quartz draft just because it's a draft. And we have these three selected. So we're going to scroll all the way back up and we're going to view the map. What we're looking for Synteny is the entire genome, the relationship that we have. That's why C-Phages wants us to compare Synteny. We look at specifically our gene 29. It doesn't have a listed protein function. So we're going to put NKF. The reason why this happens is because there are set numbers of proteins and genes that have already been put in there, and essentially what C-Phages is looking for, it's okay to put NKF if the name on Famerator isn't given. So for this one, we'll just put no known function, and then the gene that it is most similar with is Nuki. If you look at gene 29 for YUUY, it's not similar at all. It's actually way longer. So we're going to disregard YUUY for this one. In other genes, you can put it like 22, for example. You could put that one as well, but for 29, it's not. That should be it for our annotations. Again, use C-Phages for the most part. The documenting for protein coding genes, that's going to be a huge help throughout this whole process. But I hope this helps. Jackson, Tiana, do you guys have any last words? Okay.
Hope it goes well. We will still be here if you guys have questions, but this should get you started out.
Good luck.
Stop this from recording. Stop recording.